Well, good afternoon, colleagues, and welcome to the 16th Annual Massey Grand Round Symposium featuring the 3rd Annual Janet Rossant Lecture. My name is Dr. Aubie Angel, and I've had the pleasure of being part of MGR program since its start. I'm pleased to provide an update on our activities as well as some background on the MGR program. But first, it is customary that Principal Nathalie Rosiers give the land acknowledgement declaration and some welcoming remarks. Please come. Dr. Gillian Hawker will be a bit delayed and she'll speak a little later.
Welcome to Massey College. It's a great pleasure for me to have you all here and also to our virtual audience as well. I want to acknowledge that Massey College is built on Indigenous lands, the lands of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. It is the treaty land of the Mississaugas of the Credit, and we want to acknowledge our duty of stewardship toward this land and the great privilege that we have to continue to do our work here. So I want to say, without further ado, how happy I am to be here with you and to be both to celebrate the Grand Rounds and also the 3rd Janet Rossant Lecture. And I had the benefit of attending the first two, and I was so impressed that I came back for a 3rd one. So I just want to say thank you again for being here at Massey and continuing the great work that you've all been doing during the last 20 months. Merci.
Massey Grand Rounds was founded by a group of junior fellows in 2006 to fill a gap in the social calendar of Massey College, specifically with respect to the specific interests of graduate students in medicine. Monthly social gatherings and talks with leaders in medicine and health sciences were arranged, and this proved to be a lively forum for the discussion of topics and issues of interest in the lives of our graduate students. The program took hold, and with the strong support of successive masters and Principal Nathalie Rosiers, who we thank, it has flourished. I'd also like to acknowledge our sustaining donors and sponsors, the Temerity Faculty of Medicine, Dean Patricia Houston and Dr. Trevor Young, and the Department of Medicine led by Dr. Gillian Hawker. Each year, I make a point of paying tribute to our founding junior fellows and MGR fellows, shown in this slide. Fiona Menzies, Martin Betts, Jay Shaw, Patrick Wong, Janice Wong, John Leary, Laurie Waters, Andrew House, and my colleague, senior fellow John Dirks. This photo enshrines our inaugural MGR gathering in the private dining room at Massey. All these alums are doing well as professionals and practicing specialists. Now, the program has evolved over the years, and now MGR Fellows organize an annual symposium of interest to a wider community, taking into account critical health issues of the day. The recent past has been a challenge with major disruptions in our gatherings because of the pandemic. In March 2020, our MGR Symposium was canceled because of the COVID-19 lockdown. In September 2020, we had a rescue event when Dr. Sonia Nand, our 2nd Rossant lecturer, was featured in a Zoom symposium on gender and ethnic variations in health risks. In March 2021, we once again had a Zoom MGR Symposium on the impacts of COVID-19 pandemic, heart and mind. Today, we celebrate our first in-person MGR Symposium in over 19 months, and we are so pleased that this could happen. As facilities opened up this summer and early fall, we began in-person gatherings of MGR and are hopeful that the continuing success of the vaccination programs will normalize our social life as we stutter through intermittent setbacks. I'd like to acknowledge the many academic leaders and scientists who have participated over the years at gatherings with the MGR Junior Fellows. The group is designated our College of Mentors. Those in blue font are new additions. Over 80% of those listed here are senior fellows at Massey College. Thus, Massey Grand Rounds can certainly be seen to foster intergenerational collaborations. Furthermore, on closer analysis of this list, it's clear that our graduate students interact with a significant number of highly accomplished educators, scientists, and academic leaders. This undoubtedly fosters high standards and values. Our junior fellows have also benefited significantly during Gairdner Week when we host a laureate for a breakfast meeting. And thanks to Janet for continuing this tradition. A recent feature of MGR Symposium is the addition of the Janet Rossant Lectureship established by Massey College and Friends of CIHR. Dr. Rossant's distinguished career as a scientist, builder, leader in medical research, and her dedicated mentorship of young scientists are recognized in this lectureship. The lectureship is designed to host a promising mid-career scientist and thereby enliven the intellectual discourse at college. And of course, Dr. Rossant is here today, and we'll hear from her later on. Welcome, Janet. Thank you for being here. And this brings us to today's star and today's program, Dr. Samira Mubareka. Samira is a brilliant scientist, credited with the isolation of SARS-CoV-2, a brilliant and significant accomplishment, collaboration with Kozak and Banerjee. And she's a triple threat, an unusual species these days: scientist, teacher, practitioner, and busy all the time. Welcome, Samira. We look forward to your talk. She is our Rossant lecturer. She will give the keynote talk today and will speak on the human, animal, and virus nexus, insight from the COVID-19 pandemic, and will be more formally introduced by Paul Chen, our MGR Co-Chair. Our program today also features two complimentary talks by two accomplished scientists and scholars who are active researchers here in Toronto on the impact and mitigation of respiratory viruses. The relationships are very interesting, looking at everybody's CV. We'll learn in a moment that Kaveh Shojania, a speaker along with Dr. McGeer, hails from Winnipeg. I hail from Winnipeg, and Samira studied in Winnipeg. It's Winnipeg, a great export. These two speakers will amplify issues related to preparedness for the post-pandemic world, and it is our good fortune to have these senior scholars in our midst to help us understand the health impacts of COVID-19. Their more complete bios will be presented by Anna Wong, MGR Co-Chair. Now, Allison McGeer will talk about mRNA viruses, planning for future pandemics, and Kaveh will talk about reshaping research priorities to match the massive crisis we face. And we look forward to today's talks. These are the sponsors again who help us immensely over the years. I draw them to your attention. I'd now like to invite the MGR Junior Fellow Co-Chairs to moderate the talks. Anna Wong is a JD candidate at the University of Toronto Faculty of Law and a second-year junior fellow at Massey College. She's also a research associate at WHO Collaborating Center for Governance. Anna spent this past summer working for Gavi, the Vaccine Alliance, where she supported legal operation of the COVAX facility. Paul Chen is a PhD student in the Department of Chemical Engineering and Applied Chemistry and a third-year junior fellow at Massey College. And since early March 2020, Paul has investigated the factors that mediate super spreading in the COVID-19 pandemic. I invite the co-chairs to take over.
Hi, everyone. Welcome to the MGR Symposium this year. My name is Paul Chen. I'm one of two co-chairs for this year's MGR group. And I also have the fortunate opportunity to be able to introduce our first introducer for our first speaker. We wanted to invite one of our former co-chairs, Dr. Arundhati Roy, to participate in this event. Not only did they have the misfortune of not being able to have an in-person symposium last year, but perhaps more interestingly, Dr. Roy was actually one of the nominators for Dr. Mubareka to receive the Janet Rossant Lectureship. So to that note, Dr. Roy is a third-year junior fellow at Massey College and recently defended her PhD in chemical and biomedical engineering, where she studied how to grow kidney tissues in the lab using scaffolds with fractal-shaped cues. She's interested in the clinical and diagnostic applications of tissue engineering, where perhaps one day a patient's own cells could be grown in the lab and used to study their unique disease progression. Dr. Roy?
Thank you, Paul, and thank you, everyone. It is my pleasure to introduce this year's Janet Rossant lecturer, Dr. Samira Mubareka, clinician scientist and assistant professor who subspecializes in infectious diseases and medical microbiology at the Sunnybrook Health Sciences Center and at the University of Toronto. In the early days of the pandemic, Dr. Mubareka and colleagues isolated the SARS-CoV-2 virus in a level 3 containment facility. It is now the principal source of SARS-CoV-2 to many academic laboratories across the country. Dr. Mubareka serves on the Chief Science Advisor of Canada's COVID-19 Expert Panel and on the Implementation Committee of the Canada COVID-19 Genomics Network, the CanCoGen Viral Sequencing Project. She is also a member of the Ontario COVID-19 Science Advisory Table. In 2020, Dr. Mubareka formed the Sunnybrook Translational Research Program for Emerging and Respiratory Viruses to focus on the viral genomics transmission and the development of medical countermeasures from a One Health perspective. Thank you so much, Dr. Mubareka, for being with us today, and please join us.
So I'm very, very grateful for this opportunity. I have to say that this is a particularly unique opportunity because this is the first presentation that I've done in person since the beginning of the pandemic. And so I'm very grateful to Massey College, to Friends of CIHR, and all the other contributors and supporters for this effort. Merci beaucoup tout le monde. So I'll be talking to you about the human, animal, and virus nexus for this particular pandemic, which actually marks the first coronavirus pandemic that we know of, if we think about it. So previous high-consequence coronaviruses that have emerged really didn't cause a pandemic. This, in fact, was on a scale beyond what anyone expected. And I think one of the important aspects that we can't overlook here is potentially how it started and how we can derive lessons on how to prevent the next one. But before I get into that, I really want to highlight who actually does the real work. I mean, it's the highly qualified personnel across this entire campus, and the same thing across all the universities here in Canada and globally. It's the techs, it's the undergrad students, it's the postdocs, it's the graduate students, it's the research associates. Without them, we would have no data, no analysis, no impact. So I want to say thank you. They really stepped up to the plate. They were intrepid and unflappable. So I'm very, very grateful to them. So the overview for today is pretty straightforward. We're going to start at a very macroscopic level in the wild and talk a little bit about viral ecology and viral spillover and viral habitat. And then we'll talk about spillover into humans and about the virus itself. And then we'll go back to the wild and talk a little bit about what we call reverse zoonoses. So that's when zoonotic pathogens that transmit between animals can actually go from animal to human, but reverse zoonoses, human to animal. And we've done that incredibly effectively with SARS coronavirus too. So that's where I want to start, is really at that human-animal interface. One of the key features here that I want to emphasize from the very beginning is that nature is not a threat. People think about natural reservoirs, rightfully so, for a number of different human pathogens. But really, the opposite is true. We are the threat to nature, and it's really human activity that drives a lot of the spillover from animal reservoirs into humans. So I think it's really important up front that we recognize our role in that, because if we don't, it'll be very difficult to prevent the next pandemic. So there are very complex systems involved, and we'll talk a little bit about them, but it's always nice to maybe start with some possible solutions. And one approach is what we call the One Health approach. Some of you may have heard about it. It's often conventionally described as human, animal, environmental, or ecological health, and an approach that embraces all of these. But I think many people recognize that it's more complex than that. It's not just about the science and the biology, although we often start there, and we can't really go anywhere without that. That forms the basis of many things in the One Health approach. But unless you actually involve social sciences and law and governance, policy, you'll really hit major barriers. So we need all of these to work together. You don't have to have all of these aspects involved for every project, every solution. But a multidisciplinary approach that takes multiple perspectives is really critical. So let's start off with a bird's eye view here of the bigger picture. How do humans and animals interface? There's so many different ways. So you can see there are a couple of examples of very altruistic, caring relationships. There are also multiple examples of more nefarious relationships. So, for example, on my far right, I guess that would be your left, illegal and legal wildlife trade, urbanization. So you can see these little burrowing owls right next to a convertible, and then industrialization as well. And when you have things like urbanization and industrialization, you create these large edge effects. So for example, this picture here, where you have a large area that abuts a wildlife, that brings both wildlife and very, very densely populated areas in contact, that is optimal for pathogen spillover. So edge effects are really important. And then of course there are multiple other ways, either through our companion animals or directly through livestock, for example, that we interact with humans. And this is probably, with animals. This is probably the most relevant here, is in the agricultural setting or with livestock. Some of it's recreational. If you're into goat yoga, apparently there's also llama yoga available. And then also interactions between children and pets. It's always a good thing, right? But it's also a potential risk. Those are probably minor risks compared to what's happening in our food supply. So over there you have a picture of very intensified farming. That's just one pig farm. And then also we have substantial interfaces in live or wet markets that are all over the world. And the issue with live or wet markets is that you have animals of varying species coming from different geographic areas into one very densely packed place. And along with that, a public. So this is an example from Belize, where we've been down to look at bats with bat biology colleagues. And before this was all forest. On a return visit, it was obvious that there had been a substantial clear cut. And the reason for the clear cut was for farming, specifically cattle farming. So on the one hand, all the bats that roost in trees now have lost their habitat. So that's an issue, right? Because this particular area is very rich in bat diversity. At any given time, there could be dozens, if not up to 100 different species of bats. If they roost in trees, habitat is destroyed. Now, we have cattle, which makes some bats quite happy, because if you're a hematophagous bat, so these are bats that drink blood, vampire bats, Desmodus rotundus, you are thrilled because you don't have to go very far to forage for your food. This is really fast food, special delivery for a vampire bat. So they really just bite and then lap up the blood. And so they've really thrived. And the problem with this is bovine rabies. So since 2016, there have been multiple alerts about bovine rabies in Orange Walk County, which is where these pictures were taken, as well as other areas in Belize. So that's a really concrete example about how human activity can drive zoonotic spillover. Another important example is Rift Valley fever. And the reason I'm highlighting Rift Valley is because it's transmitted by a vector. So that vector is a mosquito. And the reason mosquitoes are important as a vector is because of how susceptible they are to changes in climate. And I don't mean that in that they're vulnerable. In fact, they tend to thrive in climate change. So their abundance and their range can really expand as the climate warms up. So this is really concerning when you have a virus like Rift Valley fever that can cause abortion storms in ruminants and also really severe illness in humans. Other things also drive this. Armed conflict compromises public health programs and livestock health programs, and also the livestock trade enables the spread of these viruses. So all of this was going on before the pandemic, but it was really the pandemic that triggered at least a call to action. I don't know that we've seen much action yet, but certainly there's been a call to action. So there's been a lot of discussion, the G7, the G20, around One Health. And finally, there's a commitment to operationalizing One Health as an approach to try and find solutions to mitigate further pandemic spread. So this is oversimplified, three different stages of pandemic emergence. Obviously we're at stage 3 for this particular coronavirus, and everyone's in a panic. We're spending a lot of money. We're all hands on deck. And what we're doing now, we're at the stage where we're trying to mitigate impact. But if we could go back and actually try and prevent global emergence, and ideally even prevent pre-emergence, so spillover, that would be ideal. But the problem is that section there, the wildlife or the sylvatic section, is probably the most understudied and undersurveyed group or population of all. So obviously in humans there are pretty robust surveillance programs for any number of pathogens. For livestock, because of their economic importance, there tends to be more. But for wildlife, it's very limited. Maybe some rabies surveillance, a little bit of avian flu surveillance, that's about it. And the reason that that's important is looking at these barriers here to viral spillover. There are a substantial number of them. And it really starts with the reservoir, where these viruses live. If we don't know where they live, if we don't even know what the reservoir is, how could we know anything about it and what the barriers are to spillover? So that's almost the first step. Even when you do know what the reservoir is, if we don't know anything about its population, its behavior, then that becomes a real issue. Now, if we still understand the reservoir, but we don't understand the pathogen within that reservoir, how prevalent it is, how much replication, what are the triggers for spillover, we're still sort of stuck. So it's really important that we actually look at the pathogens in the reservoir hosts. The next step is really to look at determinants of spillover, so transmission, how things persist in the environment, soil, air, and also obviously at that interface with humans, are we susceptible? Is this something that can be prevented? So fairly complex. Now there are a number of bottlenecks, some of which we understand, some of which we don't. So we know a lot more about rabies, for example. We have good surveillance programs. We've got a vaccine. We've got post-exposure prophylaxis. So bottleneck, pretty limited. Once it's in a human, you're kind of in trouble because it's almost 100% fatal. But preventing that spillover is really, really key. Ebola virus, as just another example on the opposite end, we know virtually nothing about. Just recently, I think we started to appreciate the reservoir is in fact a frugivore, so that's a bat that eats fruit. But one of the most recent outbreaks has an Ebola virus sequence exactly like a previous outbreak. The reservoir was probably a human that triggered the next outbreak. So this completely changes our paradigm or our framework for Ebola transmission. It might not have spilled over from a bat. It might be just persistence in a human and spillover. So what can we do about this? There are certain guides that are available. This is an excellent one by the FAO, OIE, and WHO, so animal, agricultural, and human health. And it's all based on essentially what we know about biology and multidisciplinary approaches using multiple tools that we've listed there. So let's focus a little bit more on coronaviruses, because that kind of gives you a big picture, a good sense of what our starting point is. So coronaviruses, I mean, you have to respect them. They're fascinating and they're incredibly versatile. They're a very, very diverse group of viruses. Unlike some of the flaviviruses genomically, which seem to align more with their vectors, you get coronaviruses that can infect any number of different species, regardless of which group they belong to. So just as an example, bats can be infected by alpha and beta coronaviruses. So SARS coronavirus 2, for example, is a beta coronavirus, clearly in bats. What little we know about coronaviruses in Canada in bats is that they're alpha coronaviruses. And there are also coronaviruses that infect avian species. Pigs also tend to get are quite susceptible to coronaviruses, and there are a number of viruses that are of agricultural importance that are infecting pigs. Now, we've all been infected by, most likely, a bat coronavirus, if we've ever been infected by one of the seasonal coronaviruses like 2290 or a bovine coronavirus. It's just that the spillover happened hundreds of years ago. Probably didn't cause a pandemic, or at least not that we know of. It probably just marched across the globe very slowly. With SARS coronavirus 2, we were very effective vectors for this particular pathogen. It's very tempting to talk more about bats, but this is the last slide I'll show because there's so much that could be said about them. People either love them or hate them. They're very divisive. But there's been a lot of focus on horseshoe bats around coronaviruses because most of them, this is where SARS coronavirus from 2003 and SARS coronavirus 2 was thought to spill over from. But actually, both alpha and beta coronaviruses can infect a pretty wide range of bats. So bats are they're also a very old group of mammals, and they've highly diversified over time. So really, the coronaviruses have sort of followed suit. And we still don't quite know how hibernation and migration affect coronavirus spread. But we're starting to understand at least some of the immunity. What's happening in bats is that they can tolerate the virus. So the virus can persist, but they don't necessarily die of infection the way we get a really exuberant inflammatory reaction if infected with SARS coronavirus 2, but not so much the bat. So that might be the sort of secret, or one of them, to the reservoirs. So I've told you about viral diversity. I've told you about the human-animal interface. What does it look like when we actually bring these together? And this is it's an old model, but I really like it because it still holds true. You know, if you imagine this very diverse group of mammals, bats, full of very diverse populations of viruses, coronaviruses, those coronaviruses will stay in the bats as long as we stay away from the bats, says the person who swaps bats. But once you start coming in proximity, then that's a different story. So MERS coronavirus is a good example of that. There must have been some overlap between bats and camels, and then spill over into humans, most likely through this intermediate host. So it's not always directly from the reservoir to the human. There can often be an intermediate host. So this spillover event happened at some point. We really don't know when with SARS coronavirus 2. Certainly by 2019, we know this virus was spreading incredibly effectively between humans and landed here in Toronto in January, very late January of 2020. And so this particular case, we had a fairly high index of suspicion given his epidemiology and presentation. So the diagnosis was made fairly quickly, confirmed at the National Microbiology Lab. The virus, the sample was forwarded to Vito as well, so they worked on isolating that virus. And fortunately, we had learned a lot since SARS 2003, so I think at an institutional level for this particular case, we were prepared. Ironically, we also had a high consequence pathogen exercise that was planned, I think the week after this patient arrived, that we had to cancel because we actually had a high consequence pathogen exercise. But it did help to prepare us for said patient. But I don't think we were ready at all for the scale of what happened after that. So we were very fortunate. Before this pandemic, I could count on one hand the number of people in Canada who could work with high consequence coronaviruses. So that's another issue too, is really who was working on coronaviruses before this pandemic, despite all the warnings with SARS and MERS, et cetera. And one of those people we were very lucky was at McMaster, so Aaron J. Banerjee. So he moved to Toronto, actually moved into my basement, this was before the lockdown, and started working on isolating the virus from patient samples. And to do that, you have to use cell culture systems because viruses don't replicate outside the cell. And you can see that on the far left, mock infected cells, they look really nice and clean, nice tight junctions, and very disrupted if inoculated with a patient sample, and the same thing reproduced when you re-inoculated. Of course, we can't assume this is a virus, so we had to do PCR and sequence the virus to confirm. But I have to say, as old school as it is, there was nothing more satisfying than seeing a picture of it. You can see, well, it might be hard from the back there, but there is a corona around that that viral envelope that consists of the spike protein, and that's where the name of the virus, coronavirus, is derived from. So all of this was done here at the University of Toronto in collaboration with many, many people, and particularly here, Mario Ostrowski, we infected a number of primary human immune cells, and none of them led to a productive infection that replicated outside the cell, or I shouldn't say replicated, but that shed virus outside the cell, but certainly inside CD4 cells, T cells, we were able to show that there was replicating virus. And since then, thanks to Dr. McGeer, we've gone on to isolate a pretty substantial number of patient isolates from a wide range of different lineages and variants, including the variants of concern, so that we've been able to continue our work. So I've already mentioned and highlighted how diverse these viruses are. Why is that the case? How do they gain in diversity? So there are two main mechanisms. One is single point mutations. You've probably heard of many of them when we talk about the variants of concern. And then there's also recombination. So it's sort of when the viral genome splices itself with another viral genome from a different coronavirus. And for that to happen, they both have to be in the same cell. So it's co-infection. And I'm not showing the data here, but we actually ran a thought experiment with Aaron J. Banerjee. What if MERS and SARS were to recombine? If they co-circulate, could there potentially be recombination? Now, luckily, they use different receptors, so you're very limited in the cell type this could happen in. They have to, it has to happen in a cell type that expresses both receptors. But who knows whether or not that could be a possibility? Hopefully, highly unlikely. Obviously, that's not something that we could test experimentally. Now, if we think about SARS coronavirus and where it lands in terms of its phylogeny, it's very closely related to SARS, obviously. So it's a sarbecovirus, more distantly related to MERS, Merbecoviruses. And it was very fortunate that early on, the Chinese government released the whole genome sequences of the viruses from those patients who had severe pneumonia in late 2019. That was important for a couple of reasons. A, we knew what we were dealing with, how closely it was related to other coronaviruses, and also just to develop diagnostics. You have to have the sequence to develop a test. So some of those early pieces of data were very valuable. It also helped to try and figure out, well, where could this have potentially come from? There's a very, very close relationship with other bat coronaviruses. But if you look at, for example, that where it says loop, that's part of the S1 protein, the spike protein. And in that small area, it looks a lot more like a pangolin coronavirus than it does a bat coronavirus. So that suggests that there may have been recombination. I think that's where the theory where pangolins might have been an intermediate host came up. I'm not sure I'm entirely convinced of that, but certainly a possibility. So knowing what the genome of the virus is can lead you to many, many different clues. We won't talk about others that probably contribute to the virulence of SARS coronavirus, but all of these factors are really important in terms of doing surveillance for new coronaviruses. Now looking more at a population level among humans with Dr. McGeer and Ontario Genomics, we did a point prevalence study. So this is where we sequenced about 950 viruses from patients in Ontario from one given day. We got as many as we could together, and the ones that were technically feasible to sequence we did. And these are from before the emergence of variants of concern, so early wave 2. And you can see here that there is a pretty wide range of variants that are involved. So none of these are variants of concern, but there's so even in humans, we're seeing the virus diversify. And if you compare the point prevalence to the first wave, there are some differences as well. First wave, there seemed to be a few peppered here and there, probably a lot of importations, and then a few settled into the community and made themselves at home. The other thing we started looking at were some of these particular point mutations. We didn't have any variants of concern, but there's evidence of what we call convergent evolution. So multiple strains or lineages of viruses that select for the same mutations, even though they're not related in any way. And we started seeing that. For example, the SQ677 that at the end there, that was evidence that things were starting to pick up. The other thing that we were able to look at is just geographic distribution of some of these lineages. Not surprisingly, and I know this is quite small, it's probably difficult for some to see, but in more populated urban areas, you saw a much broader range of viruses being transmitted. And again, this is just from one particular day. And this is work led by Pramit Seth, also earlier on in the pandemic, showing how much diversity there was among SARS coronaviruses during the first wave. So clearly, the virus was adapting. It also reflects probably multiple introductions from different places in the world. Certainly reflects that. And then a selection process ensued. So now we're facing this new threat, variants of concern, which has now become a bit of a alphanumeric soup if you think of how we've labeled them. And a lot of these changes we've really focused on in the spike protein. And the reason that's important is because that's the part of the virus that actually binds to host cell receptors. It harbors antigenic sites, so that's when we make antibodies, we make it against parts of the spike protein and also other parts of the virus as well. And it's really critical for entry into the host cell. So anything that changes around those key points, the receptor binding domain, or the N-terminal domain where there are a lot of antigenic sites, or the polybasic cleavage site, can potentially confer enhanced virulence, and we've certainly seen them confer enhanced transmission. So this is really where we are now with variants of concern. You can see that the third wave was largely dominated by B117, the alpha variant, that spilled over and drove a lot of viral activity last spring. And now it's dominated by the Delta variant. The Delta variant is in red, and the orange is mutation not detected. But to be honest, that's because we stopped looking because they're all Delta variants. So really, most of what's happening is Delta variant or some type of sublineage driven. And we're now apparently re-entering the fourth wave again. I didn't know you could do that. I didn't know you could re-enter a wave, but apparently we are. One of the other things that we've been doing is trying to determine what is the relevance of some of these changes. And so we've done a number of different things, and I just want to highlight this work because it's near and dear to my heart, looking at transmission. So for this, we use an animal model, a hamster model. And this is a contact model, so not surprisingly, if you put an infected hamster in the same cage with an uninfected hamster, the uninfected hamster gets infected. So it's a great model, but if 100% are getting infected, it doesn't really help you determine if something's more infectious. There are other things you can potentially look at, like this is in TCID50s reflect how much infectious virus there is. You can look at that. But what we decided to do is really try and look at what we're calling aerosol transmission. So these are where the cages are divided, the hamsters are not in direct contact, so there's just air passing between them. And we're comparing, for example, a more parental viral strain with something like B117. So we're seeing around the same rate, but much earlier and more robust viral replication. So obviously this work is ongoing, and we're doing the same for the other variants of concern as well. Okay, so as I promised, we'll go back to the wild and talk a little bit about reverse zoonosis just in the last few minutes. We've all heard that mink are susceptible. There have been multiple mink farms that have had outbreaks in Europe and also here in Canada and BC. But what I found really distressing was the news in the last few weeks that deer are now have been shown to be infected in the wild, which is a significant concern because once it's in the wild, there's I think many of us had ruled out the likelihood of eradication anyway, but it cannot be eradicated once it's spilled over into an animal reservoir, and there's always the chance of further viral adaptation and reemergence and undermining medical countermeasures. So this is just one of several studies that have come out recently, but the concern here is it's not just serology. This is actual viral RNA in these deer, about 35% of the ones that they sampled in Ohio, and in some groups it was actually even higher than that. It also looks like there were multiple introductions. There are some clusters, so either a very common exposure or they're transmitting to each other, and then a few singleton exposures as well. So that to me spells trouble. We're doing screening for SARS coronavirus 2 in wildlife, and we've been working with some really great wildlife biologists. If you want to restore your faith in humanity, collaborate with a wildlife biologist. The Canadian Wildlife Health Cooperative, the Ministry of Natural Resources, the National Centre for Foreign Animal Diseases, people at Guelph and Trent, really been amazing and worked together to collect samples and do the testing. So we focused on what we call synanthropes. So these are peri-domestic animals that we're probably in contact with. And also because of the farmed mink, I think, we've also looked at wild mink. Though I have to say the wild mink were really what we call convenience samples. So they're coming from trappers as opposed to the raccoons and skunks who were actually swabbed sort of in a more prospective manner. And not surprisingly, most of our animals did come from southern Ontario because that's where we're anticipating spillover to have happened, but that's also where a lot of surveillance is done. And this is why the importance of surveillance systems is highlighted, because a lot of the skunks and raccoons actually came from rabies surveillance programs. So you can leverage different surveillance programs to look for novel pathogens. Obviously, since it hasn't been in the news, they've all been negative, fortunately. And I have to say, I snuck in a couple of slides just on highly pathogenic avian influenza virus, not to cause alarm, but just to remind us that we cannot be complacent. We're seeing increased signals just since May, which is quite concerning, of H5NX. So there are multiple subtypes that are being transmitted, particularly around animals. So last week there was a notification from OFFLU, a couple weeks ago from the FAO. And then unfortunately, there's also been a spillover into carnivores in the Netherlands. So foxes have become, so an avian influenza virus to spill over into a carnivore is a big deal. They're usually not susceptible. So that's quite concerning. We have done some surveillance in swine and derived some genomes to have a look and determine whether or not certain environmental sampling approaches are helpful. I'll maybe go just to the next slide in the interest of time, because I think it's probably even more relevant. So this is a project led by the National Centre for Foreign Animal Diseases with their partners in Ghana to do live market, so wet market sampling, and also backyard farm sampling. And our part is actually quite small in it. It's this little bioaerosol sampler to look at aerosols, and we're helping them in a couple of other ways. But this just goes to show, so Ghana had a few highly pathogenic influenza outbreaks, and really it was pretty obvious, at least 5% of birds that were sampled were PCR positive. And not surprisingly, they're H5N1, the clade that's been circulating, that the alerts have been issued 2344B. So again, there's been neglect of some other surveillance programs because everyone's so focused on SARS coronavirus, but it's really critical that we not ignore that. So just to summarize, there's definitely a role for surveillance, especially at the human-animal interface, to understand ecology, pathogen characteristics and risk, and taking a One Health approach is one way to do this. We really need a radical paradigm shift in how we approach this. I think if we think of health the way we have been thinking of health, which has been primarily from a human perspective, we're missing a lot of these other pieces that is just going to lend to history repeating itself. So we really have to think about health from a much broader perspective and really start thinking about reciprocal care and how we can look after the land and everything that's on it. So we have there are a few ways that we can do that. There already is a pan-Canadian approach for wildlife health that's been agreed on between the provinces, territories, and the federal government, but it hasn't been implemented yet. There are a couple of other really important tools that could be used. We certainly have to do things better at a global level. And then don't forget the fundamentals around education and research for One Health. So I'll stop there and say thank you to many more people, not all of which are on this slide, but I'm very grateful to everyone. Some of these are projects we didn't discuss, but we're still part of our activities, our key activities over the last 2 years. Thank you.
As Vice Chair for Quality and Innovation in the University of Toronto's Department of Medicine, Dr. Shojania developed a new academic career track for faculty members called Clinicians in Quality and Innovation. More recently, Dr. Shojania has become interested in ways of achieving improvements in actual health instead of just improving healthcare by collaborating with groups addressing the social determinants of health, as well as obvious threats to health and healthcare, such as the climate crisis and systemic racism. Please join me in welcoming Dr. Shojania.
Thanks very much, and thank you for including me at this event. Before I get started, I just want to make clear, especially after that terrific scientific talk, that when I'm talking about reshaping research priorities, I'm not talking about not doing certain types of research anymore, but I am talking about the targets of those research. And there's one symptom of that. I'm surprised more people don't talk about this, but I don't know how many people realize, but Bill Gates gave the Shattuck Lecture on Pandemic Innovation and was published in the New England Journal in May 2018. And he called for widespread research into pandemics. And I actually checked just last night on PubMed. In the next 19 months until the end of 2019, there were 4 articles that mentioned the word pandemic in the New England Journal. Since January 2020, there's been 400. So it's not like we didn't, and of course in Canada, we had the whole embarrassing situation with the Public Health Intelligence Network, where we disbanded this highly regarded pandemic alert system right before the pandemic. And instead, we decide we need more antiplatelet agents and lipid-lowering things and like monoclonal antibodies for advanced cancers instead of these obvious threats to our health. So that is what I'm talking about, or plan to talk about for the next few minutes. Now, my title refers to massive crises. I think we all have an idea of what they are, but just to be clear, I'm referring not just to COVID, but the near certainty of future pandemics like it, the climate crisis and collapsing ecosystems, which, as Sam alluded to, is as important as obviously the greenhouse gases themselves, ongoing structural violence against racialized groups, worsening income inequality, which is really approaching levels last seen under feudalism, and the opioid epidemic, which in parts of Canada and the US lowered average life expectancy even before COVID did so. So these crises will worsen already existing disparities in basic health outcomes within cities and across countries, much as we saw during COVID. What do these disparities look like? Well, here's a particularly striking example I came across a couple of years ago. Many of you will recognize the London Tube map, but the stations are shown with the average life expectancy in the surrounding neighborhoods. Now, if you notice along the red line, there's stations with high life expectancies like 90, then 96 years, then it drops to 85, and the next stop in either direction is 79 years. So that's a 17-year difference in life expectancy just on 2 stations. There are almost no biomedical interventions that impact life expectancy like that, except public health and hygiene, the things we've watched be eroded over the last 75 to 100 years. Now, we don't have as striking a picture in Canada, but we have had studies showing the same things. The Code Red study in Hamilton, a similar study in Sudbury, and Winnipeg was mentioned earlier, and actually the life gap between the richest and poorest neighborhoods in Winnipeg is 18 years, and at a national level, you need to go back to 1940 to have an 18-year difference in life expectancy. So the bottom line, the Canadian Medical Association put together this graphic about 15, 20 years ago. And you may quibble with the exact percentages shown, but I don't think you can really argue with the qualitative picture that what we prioritize in healthcare or healthcare research does not reflect what makes most people sick. In fact, you'll see that biology gets only 15% and the environment 10%, although I think these days people might inflate that percentage a little bit. Not only does what we prioritize the wrong thing, but what we do actually prioritize usually doesn't work. Most of you who are in healthcare will know when you open the New England Journal or JAMA or any major journal, it's like one trial after another showing that this thing didn't work after all, or it has a tiny effect size with really high numbers needed to treat. There was actually a really elegant, cute little study in PLOS One a few years ago that looked at large NIH-funded cardiovascular trials before and after the year 2000. And they picked the year 2000 because that's when clinical trial registration became mandatory and you couldn't lie about what your primary outcome was. Before then, 50-some% of trials were positive. After, 8%. So greater than 90% of trials, and this is after massive investments in phase 1 and phase 2 trials, and many of these drugs are already out in practice, and then finally a large trial is done and shows that it doesn't work. Not only that, but the ones that do work usually have, I would say, almost homeopathic impacts. Statins, famous for lowering cholesterol, and the actual number needed to treat is that 33 to 100 patients need to take a statin for 10 years to prevent one cardiovascular death or ischemic event. And notice that's cardiovascular death, which is actually not quite the same as death. There's an implicit subjectivity there. And so I actually did look into what the longevity effect of statins are, and there was actually a couple of really elegant analyses that basically showed that the impact on longevity is 10 days if you don't have cardiovascular disease and 17 if you do. Imagine if instead of a vaccine for COVID, we spent billions of dollars a year on a medication that 33 to 100 patients needed to take for 10 years in order to prevent one COVID-related hospitalization or death. We are so used to seeing therapeutic effects reported in terms of odds ratios and relative risks, often on disease-specific endpoints, that we forget the rarity of medical interventions that materially improve the length and quality of people's lives. Here's an example to show the type of concrete choices we could make. Obviously, dementia is often in the news. Now, we could continue the ongoing quest for a magic bullet. In fact, I looked up recently, there was a paper in PubMed showing that there's 121 agents currently in clinical trials for Alzheimer's. Now, the likely future outcome is a statin equivalent. In other words, a medication that 30 to 100 patients with dementia need to take in order for one person to derive clinical benefit. Now, why would we continue to invest almost all of our research efforts into that pursuit instead of focusing on the obvious structural problems and how we care for the elderly, better models of care for caring for the elderly in community, develop and refine new models for long-term care facilities, optimal staffing levels, all the things that we saw during COVID wreaking total havoc in the nursing home care sector. We've mentioned the climate crisis a few times. Here's a concrete example related to the climate crisis. Just before COVID came out, in October of 2019 actually, this paper in the New England Journal in the top bullet, it was an amazing study. Across 652 cities, they measured ambient particulate matter, basically pollution. And they were able to show that even small increases changed all-cause mortality by 0.5% within 48 hours. Now, for those of you who are not in medicine, we almost never impact all-cause mortality, almost never. So to have in 48 hours pollution producing that effect is really staggering. And as many of you may have heard on the radio last year during wildfire season, and again this year, there were studies documenting increased 911 calls and ED visits within hours of particularly bad smoke air quality. And actually in Australia, they documented excess mortality. And for a number of years now, the WHO and the UN have had a campaign identifying pollution as causing a quarter of deaths from cardiovascular diseases and 40% from lung diseases, and actually the global mortality impact is the same as that for tobacco. Now, in that context, why would we keep researching new inhalers and even monoclonal antibodies now and then not invest anything in working with people in other sectors to improve air quality, reduce pollution, and actually prepare the healthcare system to be able to handle the surges of patients that are going to happen increasingly frequently every summer? I was actually in Winnipeg this summer during the heat dome and the wildfires visiting my mother in palliative care. Even in the palliative care unit, my eyes were stinging all day from the quality of the air. And some of you have been listening to the news today about BC. Like, it is amazing how quickly this can cause immediate, like, obviously general catastrophe, but obviously health impacts. Another thing we could do, which obviously with the public health theme of today, is instead of whether just talking about like the social determinants of health and poverty and so on, or the climate crisis, is we could just in general invest more in disease prevention. And actually there was a wonderful book by Dan Heath. He and his brother Chip are management professors. Chip and Dan Heath, it sounds like a comic book, but at Stanford and Duke. And they wrote a number of very well-regarded books called Switch and Made to Stick. And Dan Heath wrote his most recent book, Upstream, in which he wrote that the US healthcare system is designed almost almost exclusively for reaction. It functions like a giant undo button, but it's hard to find someone in the system whose job it is to answer the question, how do we make you healthier? We all agree that an ounce of prevention is worth a pound of cure, but our actions don't match our words. Now, the book is very interesting, filled with a number of inspiring examples. This one really stuck with me, and I used it when I was giving actually a talk in Chicago not long ago. So this is, and by the way, that's President Obama sitting at the bottom here in the meeting here. So this was became a very prominent, well-known intervention in certain circles. So among racialized young men in Chicago, and I'm assuming most other major American cities, homicide claims more deaths than the next 9 causes of death combined. And apparently they're not gang related. They're often disagreements that escalate quickly in the context of access to guns, drugs, and alcohol. So this intervention focuses on impulse control, conflict resolution, and future orientation, and so on. And it was developed over some years by a nonprofit, and then the crime lab at the University of Chicago got involved, helped them refine it further. And now in 2 randomized controlled trials, they've documented absolute reductions of 28 to 35% in violent crime arrests, and even increases in graduation rates. So these are obviously, we're talking about the social determinants of health. We need more research that actually addresses the social determinants of health. An example I don't show because it's such a short talk, but some of you may know that there was a randomized controlled trial published in the Journal of American Medical Association a few years ago, led by investigators from Toronto, in which they actually increased days stably housed for homeless people with mental health problems. So these are the types of things we need more research of, I would argue, in the context of a post-pandemic world. But these are socioeconomic problems, not healthcare problems. Well, maybe, but healthcare consumes 40% of provincial budgets. If we keep taking all the money and not doing anything about these socioeconomic factors, we will never improve population health. The vast majority of biomedical research is aimed at targets which have little chance of improving basic health outcomes, and will have even less chance of doing so as the consequences of stark economic deprivation and the climate crisis both worsen. I came across this quotation some years ago. Max Planck, the Nobel laureate in physics, once said, "A scientific truth doesn't triumph by convincing its opponents and making them see the light, but rather because they die, and a new generation grows up that's familiar with it." We don't have a generation to wait, though. The earth is burning. Even in wealthy countries, large segments of the population have problems that the current healthcare system are not addressed to remedy at all. And of course, in lower income countries, we see mass population displacements due to violent conflict. Lack of land, food, water, and jobs will similarly overwhelm our current approaches to humanitarian medical aid. This was a cartoon I remember seeing as a kid from the far side. The picture's pretty bleak. The world's climate is changing, the mammals are taking over, and we all have a brain the size of a walnut. We have larger brains, but it soon won't matter much if we don't start acknowledging the massive mismatch between our current research priorities and the crises we face. Even when our current approach succeeds, as with the COVID vaccines, the potential benefits are undermined by our lack of attention to the social sciences relevant to public engagement and messaging. Big pharma, big tech, oil, agribusiness, they all pay attention to and harness techniques for shaping public attitudes. We need to start doing that as well. So in conclusion, most biomedical research is not directed at the main causes of poor health. We cannot continue to prioritize research on expensive new medicines delivering marginal benefits. We need more research addressing the social determinants of health and disease prevention. And as I mentioned, COVID has highlighted the importance of the social sciences as well in shaping public opinion. And I'll just end with this passage from an essay that Arundhati Roy wrote last year in the Financial Times. Historically, pandemics have forced humans to break with the past and imagine their world anew. This one is no different. It is a portal, a gateway between one world and the next. We can choose to walk through it, dragging the carcasses of our prejudice and hatred, our avarice and data banks and dead ideas, our dead rivers and smoky skies behind us, or we can walk through lightly with little luggage, ready to imagine another world and ready to fight for it. Thank you.
In 2003, Dr. McGeer led the hospital investigation of the SARS outbreak in Toronto. She subsequently worked in the Kingdom of Saudi Arabia investigating MERS, and in Liberia during the 2014 and 2015 West African Ebola outbreak. During the COVID-19 pandemic, Dr. McGeer has led work on controlling outbreaks in long-term care homes, studied the transmission dynamics of COVID, and in collaboration with Dr. Mubareka, showed the utility of saliva testing for COVID-19 diagnoses. She served on the Influenza Working Group of Canada's National Advisory Committee on Immunization, and the Ontario Immunization Advisory Committee. And she's also a member of the Canadian COVID-19 Immunity Task Force Leadership Group, the Ontario COVID-19 Science Advisory Table, the Canadian COVID Genomics Network Steering Committee, and Canada's Chief Science Advisors Expert Panel on COVID-19. Please join me in welcoming Dr. McGeer.
Thank you. You will notice in the next few minutes that I'm not going to talk a lot about mRNA, and there's going to be some overlap with Kaveh, but I think that the overlap is overlap that bears repeating. So one of the challenges of pandemics that are hard and fast, okay, so there's a whole range of different types of pandemics, right? HIV, antimicrobial resistance, completely different pandemics, completely different sets of problems. But like influenza pandemics, COVID pandemics are hard and fast. And the problems with those are that by the time you get close to the end of them, and we've got a ways to go, and you know how you feel today you just want out, right? 6 months from now, it's going to be worse. And the history of these kind of pandemics is that when we get to the time that they have faded and we're getting back to normal, we all just want out so badly that we don't do the work that's necessary to make the changes that will make the next pandemic better. So for the next 15 minutes, I'm going to give you some of my thoughts on the things that we need to do or the things that we need to work on, along with some good things that have happened during the pandemic. Not so much because I think these are the most important things, but because I want you to start thinking about where we go from here and what we need to hold on to and change going forward. Okay, so there's some things I'm not going to talk about that because they're still up front in people's minds, not necessarily that we're going to make progress on them, okay? I'm not going to talk about inequality in global vaccines. That's not going to go away. We all know that's a problem. Hopefully we can work on it. I'm not going to talk about nursing homes as long as we're reading every day about the inquest in Quebec. That's such it is. So let's start about some things that we might think about that are, I think even now, kind of fading from people's center of attention about fixing it. So this is from the Johns Hopkins Our World in Data, amazing group of people who've been getting what data can from around the world that looks at cumulative deaths per million from COVID-19. And this, I think, is the way we see ourselves now, okay? Countries that we usually compare ourselves to, US, the United Kingdom, Northern Europe, we're doing pretty well, right? Yeah. And most people, I think, are reasonably satisfied that in Canada, we've done okay. So let me give you a slightly different view. Okay? I've taken the island nations out. Okay, island nations have an unfair advantage in the pandemic, unquestionably. So Japan, no, I took Japan out too. So no island nations here. But look at how much better we could have done. And I would put it to you that this is about what Kaveh was talking about. These countries are countries that have solid, well-resourced, adequately funded public health systems. If we had the public health system that Norway has or South Korea has, we could be there. I'm not sure we can do it. This is the one that I think we're least likely to accomplish, okay, because it's not like we haven't been here before with public health and knowing we need to fund them. But it is a critical element of moving to prevention is something that we have to do. Okay, the second thing is about mRNA. And I was on a Zoom call last week with a colleague who said, wasn't it nice that the pandemic waited until we had Zoom so that we could actually manage things? And he had a point, okay? But what was really nice was that the pandemic waited until we had mRNA vaccines, right? That's what has made all the difference. And the thing about the mRNA vaccines, it's not the mRNA vaccines. The miraculous thing about having vaccines, to my mind, is actually knowing how to get the right conformation to the spike protein to put in them. Okay? mRNA vaccines have lots of other benefits. People have been working on them, a bunch of good commercial reasons why you would want to develop mRNA for a whole bunch of purposes. But understanding the spike protein and its conformation, that was Barney Graham and Jason Walsh. Your life and mine have been preserved by two guys who just happened to work at the NIH. And what's really cool about what they discovered, so Barney Graham was driven by the fact that he was one of the people who worked on the original RSV vaccine for kids. Many viruses, HIV, RSV, SARS coronavirus, have the equivalent to the spike protein in the SARS coronavirus that's responsible for binding. And all of these proteins, it turns out, do calisthenics, all right? When you formalin inactivate the virus, when you kill it for any way, when you isolate protein from the virus, what you get is those spike proteins in what's called post-fusion conformation, okay? So spike proteins that sit on the surface are all curled up. When they go to bind, they unfold, and then they stretch. And eventually they fold over so as to allow viral RNA to enter. And what you get, when they formalin inactivate the RSV vaccine, they got post-fusion conformation. And you can appreciate the fact that that has very different antigenic sites on it. What your immune system sees is very different when you're looking at something that's all stretched out versus when you're looking at something that's folded in on itself. The antibodies that you make to post-fusion are not neutralizing. They actually enhance repeated infection. And 2 kids in the RSV vaccine trial in the 1960s died in that trial because they got antibody-enhanced response to a repeated RSV infection afterwards, which is why we only just this year have an RSV vaccine 40 years later. So when Walsh moved to Barney Graham's lab, he was actually working on HIV. And he wanted to get the structure of the glycoprotein on the surface of HIV that binds so as to work on HIV vaccines. And he couldn't make it happen. And Barney eventually talked him into working on RSV instead, just because Barney felt badly about the RSV and he really wanted to understand what had happened. And 10 years later, he had the structure outlined, and they figured out, by a process of elimination, how to make a series of mutations at the base that would lock spike proteins into pre-fusion conformation. And every single every single vaccine, never mind mRNA vaccines, every single vaccine that we're making against COVID has those 2 mutations that lock spike protein into the pre-fusion conformation so you and I get the right antibodies and we get protected from disease. And that's a miracle. And the message from it to me is, we don't know where basic research is going to take us. We can't predict ahead of time which piece of that basic research is going to benefit us. And we are not doing well in Canada, compared to the rest of the world, in funding basic research. But basic research is what makes the miracles when we have pandemics. So I'm hoping that we'll get some degree of reminder of that and get ourselves set to do more of it in the future. Okay. And this is structural inequality. And one of the amazing things about the analysis of data from this pandemic in Ontario is that Toronto Public Health has been willing to work really hard at looking at structural inequalities in the city, and that a group of investigators, far too numerous to name, okay, but Jeff Kwong, Beata Sander, Sharmistra Mishra, and on, have been working on looking at heterogeneity in COVID-19 and COVID-19 outcomes. And the result of that is that you see exactly what Kaveh showed you for so many other outcomes, which is that the most important thing that predicts whether you die from whether you have been at risk of dying from COVID-19 in the last 18 months is how much money you make. And all of us in this room are pretty comfortable and are likely to go on, but we have allowed dramatic inequalities to build, including in Toronto, where we tend to think that doesn't happen, right? We see ourselves as nice people, kind people. But the degree of inequality that we have in Toronto is something that we need to think really seriously about. And whether we are willing to continue to tolerate that, or whether we need to change it going forward. And lastly, I want to talk about data and information sharing. So one of the other good things about the pandemic is that all sorts of people have been very good at putting data together and creating information for us. So I'm just giving you one example from the Science Table Secretariat that updates daily your risk of getting COVID and being in the hospital and being in the ICU by whether you're vaccinated or unvaccinated. Really helpful, powerful messages for people about the benefit of vaccination, and done on an ongoing basis because the ministry has really put a lot of work into trying to get us data that will help us understand what's going on and move it forward. And here's another tremendous example. This is Jeff Kwong's CERN work, in which we now have, because the ministry created a vaccine registry, okay, first in Canada, okay? It's another miracle. Jeff Kwong can actually give us vaccine efficacy over time in all sorts of detail in real time for us in Ontario. Now here's the but, okay? We can only do it for Ontario, okay? We can't share data between provinces, complete barriers between provinces. And there's a great deal of other data that we can't have, and a large number of things we can't share. And to me, there is a huge discrepancy between the way we have built privacy structures and the way people want privacy structures. One of the things that's been really interesting to me is that because I lived through SARS and we had no mechanism for sharing anything during SARS, when COVID started, we set up a program that was deliberately designed to get samples and data to people. Don't care who they are. If you need data to, I don't know, develop tests, do analyses, isolate viruses, that's what we're here for. And so one of the things we did was that every patient that we've enrolled in any of our studies gets asked in pretty threatening language to share their data and samples with anybody who asks for it. So pharmaceutical companies, people from other countries, there's some very, to my mind, threatening language that the REB requires, totally reasonably, we've all agreed to it, about the fact that, you know, data protection is not good in other countries and pharmaceutical companies may make money from your samples and you won't get any of it. All of that's in our consent forms. 2,000 people we've consented. Not a single one has said no to that component, right? There's a list of components, and this is one you get to choose yay or nay. At the beginning, I was actually too scared to ask some groups of people about this because I thought it would just really offend people and it might make them go away from the whole study. Nobody cares, okay? Every single person in this has said, yep, that's fine, not a problem. And yet, our organizational structure tells us we can't do that. Our organizational structure says we have to have formalized data sharing agreements with this huge bureaucracy around how we share data that really seriously interferes with how we get data to people during pandemics and what we can do, and never mind during pandemics, for all the other things that we need to be doing. And so this is my last plea to people, that we think about how we want to use the data that we can harness about people that can be used to move people's health forward, and see what we can do about the serious systemic barriers that we have in research in Canada to be able to take advantage of that. And with that, I will say thank you.